which basically functions to make a new ATP molecule. And here's succinyl-CoA, which is the product that comes off of those decarboxylation reactions. And so just like acetyl-CoA is formed in PDH, succinyl-CoA is formed in alpha-ketoglutarate dehydrogenase complex. So alpha-ketoglutarate is like pyruvate, except instead of 3 carbons, it's got 5, and then you split off a carbon, CO2, to make succinyl, 4 carbons, but it's linked to a CoA, so it's high energy. Okay. So the way this enzyme works is it facilitates attack of phosphate, inorganic phosphate, release of CoA to form a succinyl phosphate intermediate. This is an acyl phosphate. You know that's a high energy linkage because we found a compound in glycolysis and talked about it earlier. That's a very high energy linkage. A histidine on the enzyme attacks the phosphate group of it to release succinate. Succinate is the compound that is then racemized with respect to the C14. Right? And now you have a phosphorylated histidine. Right? And so we've preserved this high energy linkage. First it's here. Now it's in this bond, acyl phosphate. Now it's in this bond, phosphorylated histidine. This is still a high energy bond. And now it's transferred to GDP to make GTP. And then there are sets of enzymes in the cell. They're called NDP kinases, which basically shuttle around the terminal phosphates among NDPs and NTPs. So GTP plus ADP will make you ATP. Right. There's also uses in the cell for GTP, so maybe we'll keep it as GTP. If we want ATP, you can get there with delta G of 0. As long as you're at the same high energy level, you can come in. Okay? So remember when you're here in this cycle, right? There's only a few things to say about these last steps, which we can cover next time. But when you're here, you've lost that racemization, right? And then you have dehydrogenase, addition of water, and another dehydrogenase, and here you're generating your reduced cofactors. The amount of energy you can get out of this is listed here. So right now, this is just arithmetic. Glycolysis, invest in ATP, -1, invest another one, -1. Make 2 NADHs per glucose, so this is per glucose. Aerobic oxidation of glucose by glycolysis, PDC, TCA, and oxidative phosphorylation. Notice that here it says 3 or 5. We will resolve 3 versus 5. NADH can get you 5. There are times when some of the reducing power is lost. If it has to come from the cytoplasm and get to the inner mitochondrial membrane, maybe it doesn't get there. Sometimes it does, sometimes it doesn't. Otherwise, NADH is worth 5. Right? FADH2 is worth 3. And we will explain this when we get to oxidative phosphorylation. We get 2 more here, the substrate level phosphorylations, the 2 in glycolysis, the 1 in the TCA cycle. Right? Here's pyruvate going to acetyl-CoA. That's from the PDH right here. So you add them up. Right? If it was standard state, right, this would be 32. Let's say it's 32. It would be 976 kilojoules per mole. But in the cell, it's not 32. It's more like 50 something. So the amount of free energy you can get out of a glucose molecule as compared to the amount that's locked into glucose, and we talked about that from before, it's 2840. Under cellular conditions, the efficiency is something more like 65%. If you fully convert glucose to CO2. So that's a lot better efficiency than you can get with your internal combustion engine in the car. And it's done by virtue in lots of little steps, conserving ATP energy along the way, usually as reduced cofactors, but ultimately using those to make ATP. Okay, remember there's a quiz. It's due Friday. It's online, so it's available. And I'll see you Friday.